chapter 15 is going to be immunization, providing immunoprotection through therapy, the different vaccines we use and things like that. So we have specific immunities. That's our adaptive line of defense. Remember, we have the first two lines of defense we talked about last time. First line of defense is your physical barrier, like your skin. Second line is your inflammation once it gets past the skin. The third line is our immune system. Okay, that's what once things get in, that's what it all boils down to. So the third line of defense, okay, is our adaptive or acquired immunity. Remember, adaptive is what we've learned, like we've learned to be that certain things are foreign, or acquired is what we were born with, the ability to just find things as foreign to our bodies. Okay, so the product of B and T lymphocytes is a dual system. We use B and T lymphocytes. We talked about it a little bit. Immunocompetence. Immunocompetence means is the ability of the body to interact with a wide spectrum of foreign substances. Immunocompetence is when a cell becomes active. Remember, we talked about B and T cells both being formed in the bone marrow. The T cells migrate to the thymus, where they become immunocompetent. They mature there and become what's called immunocompetent, able to recognize foreign substances. Okay, so that's what immunocompetence is. B cells become immunocompetent in the bone marrow. T cells in the thymus. And antigens. Antigens are the molecules that stimulate a response. Those are the things that we recognize as foreign, are antigens. Okay? So two features characterize a specific immunity. We have specificity, which means it only attacks things that are foreign. And we remember those things. It's very specific what a B and T cell will attack, and it has memory. Once it's exposed to it, it remembers it for later on. That's the whole base for behind a vaccine, that the body remembers it and it can respond quicker, doesn't have to reproduce and produce all the B and T cells for that one antigen anymore because it remembers it. And we already have immunity built in. So specificity is antibodies are produced. They function only against the antigen that were produced in response to the attack. In memory, lymphocytes are programmed to recall their first encounter with an antigen and respond rapidly to subsequent encounters. They don't have to remake all the T cells that are specific to that antigen. Everything's already in progress. So our overall view of our specific human responses, immune responses, you got separate but related activities of specific immune responses. You got development and differentiation of the immune system. We start with stem cells and we develop into the T and B cells and the specific type of antibody that we want to produce. Lymphocytes are your white blood cells, and antigen processing. Okay? We identify the antigen by certain lymphocytes, certain white blood cells attacking it or attaching to it. The cooperation between these lymphocytes during the antigen presentation. So we present it to the white blood cells, and then it determines what response the body is going to have. The B lymphocytes and the production and actions of the antibodies. And then once the B lymphocytes are made for that and respond to it, then we get the T lymphocytes, which recognize it and produce the appropriate response. If it's a cytotoxic T cell, it'll start to kill the antigen. Okay? So this is the specific immune response. So the first one is an antigen independent development. This is our T cell. Remember, here's your mature stem cell, and matures into either a T cell line or a B cell line. T cells go from the bone marrow to the thymus, and if the thymus, they become immunocompetent and go into the lymph nodes. The B cells, they stay in the bone marrow, become immunocompetent in the bone marrow, and then proceed to the lymphocytes, into the lymph nodes. Okay? So you get migration to an establishment of T and B cells in lymphoid organs, like your lymph nodes, your spleen, which is the largest lymphoid organ, and the MALT. Okay. Development of the lymphocyte system. Okay. Lymphocytes arise from the same stem cell. The one before here, this is the same stem cell. Both B and T cells arise from that same stem cell, but differentiate into two distinct pathways. Okay, T cells mature in the thymus. That's where they become immunocompetent, and B cells mature in the bone marrow. Once they're released, the mature cells settle in lymphoid organs. Okay, like in the spleen, lymph nodes, or the MALT, mucus-associated lymphoid tissue. Okay, and these once they settle in these organs, this is where they serve as a constant attack force for infectious agents. That's where they hang out and wait to be called into reserve to respond to this assault on the body. Okay, so then we've got from here, we got the antigen contact. Okay, we remember the dendrite cells have all these branches on it, and they can trap the antigen, and then the helper T cells respond by binding to that. Okay, they present the antigen to the B cells, B cell receptors. Okay, if the B cells recognize it as foreign, then it differentiates the B cells into memory B cells and plasma cells that can go into the bloodstream and move towards the attacking system. Okay, once these cells are produced, it goes on to the T cells. You have an activated T cell when it binds to the antigen. Okay, cytokines are produced. These cytokines are going to receptors that differentiate these memory T cells, okay, into helper T cells and cytotoxic T cells. The helper T cells are used up right here to pass it on to the B cells to identify it. The cytotoxic T cells are used to kill or destroy the antigen. Okay, the B cells form your memory B cells, which stay in the memory so that you don't have to reproduce all this stuff a second time once you're exposed to the same antigen. So this the response, you have the contact with the antigen. So the antigens get past the first and secondary defenses, gets exposed to the lymphoid tissues, lymph nodes. Then you have contact with the antigen, and we get presentation by the antigen. Remember, we talked about antigen presenting cells last chapter. Okay, so foreign cells have molecules on them called antigens. Okay, they're recognized and engulfed by these antigen presenting cells, like the dendritic cells, which are all those branches that trap them. Okay, T helper cells receive the processed antigen from the antigen presenting cells and go on to activate the B and other T cells. Okay, the B cells form your memory cells, T cells form your cytotoxic cells, and then your helper T cells also. Okay, you get activation of the T cells, and then you get a T cell response. So an activated T cell, again, forms your memory cells, so you remember this antigen for later on. So you don't go through all this process later on, you just respond to it and it responds quickly. It differentiates into helper T cells, okay, and into cytotoxic T cells. So T cell immunity, okay, it's termed cell-mediated because the whole T cell acts directly to destroy the microbes rather than secreting molecules in body fluids. So the T cell acts as a macrophage, opens up and actually destroys the microbe or the invading pathogen, which is your antigen. Your B cell response, remember the B cell was activated by the helper T cells, okay, that bond with the antigen. And then the decoys goes through a spurt of cell division, producing our memory cells, okay? Provide a rapid recall of that antigen, and it circulates through the plasma. So if it finds it, it can duplicate it right on the spot. And they secrete proteins called antibodies. These antibodies are our memory of what is not foreign, not self, that's what's foreign to our bodies. So the B cells and T cells are tied together. You have a humoral immunity. Okay? Antibodies circulate in the fluid once they're made from the B cells. They circulate in the blood. They circulate in the extracellular fluid, and they circulate in the lymph. And these provide humoral immunity. Okay? So the antibody reacts specifically with the antigen at the second exposure and mark it for an enhanced response. So you cannot get humoral immunity, you cannot get antibodies until you've been exposed to it first. Okay, the first time you're exposed to something, you cannot get a response. You have to build up that response. Okay? Once the second time comes around, you already have that information in the memory B cells, and then you get your quick response. Okay. Cell receptors or markers confer specific identity of a cell. On the cell's membrane, we always have these cell receptors or markers. We can identify whether a cell is foreign or whether a cell is self. Okay. Major functions of these receptors are to perceive and attach to non-self or foreign molecules, promote the recognition of self molecules. Because you got to remember this immune system, your immune system is designed to kill what's not supposed to be there. So in order to do that, you have to identify what's self and what's non-self. You don't want to start killing your molecules that are your body. Okay, so you have to recognize the difference between self and non-self molecules. And for that, we use these receptors. Receptors are also used to receive and transmit chemical messages among other cells of the system, so cells can communicate with one another through chemical messengers, and it aids in cellular development. This is what functions of the receptors on the outside of the cell membrane. Sets of the cell surface protein, these receptors, okay, are essential for the acquired immune system in the recognition of self and rejection of foreign molecules. When we talk about the MHC molecules that are presented with the B cells, okay, are found in all cells except the red blood cells. So red blood cells have no markers on the outside. Okay, red blood cells are ones that do not have a nucleus. When red blood cells lose their nucleus, they lose their mitochondria, they lose all the cell organelles, and all they are basically is a sac, it's a biconcave sac that holds hemoglobin, so it can bind oxygen and carbon dioxide too. Okay, that's all they're designed to do is carry oxygen. The carbon dioxide binds to the CO2 binds at a separate side of the hemoglobin, okay, so it doesn't interfere with the oxygen delivering system of the hemoglobin. So red blood cells are anuclear, and they're the only ones, our cells, that do not have these HMC molecules, receptors on the outside of their membrane. HMC is also known as the human leukocyte antigen, HLA system. The MHC gene, divided into two main groups. You have class 1 and class 2. Okay. Class 1, MHC, the receptors. These are genes that code for markers that display unique characteristics of self, now recognition of self molecules and regulation of immune reactions. So it identifies which one is self so you don't respond to it. It's required for T lymphocytes to interact with pathogens. Okay, the class 2 HMC receptors found on the membranes are genes code for immune regulatory receptors that are found on antigen presenting cells like macrophages, dendritic cells, and B cell, which identified foreign or non-self cells. Okay, they're involved in presenting antigens to the T cells, because once they find it as foreign, they present it to the T cells, then the T cell responds. It makes more helper T cells to increase response, and it makes cytotoxic T cells to kill the antigen. And this is what MHCs look like. Here's the outside of the cell. This is your cell membrane, and these are peptides. Peptides are just proteins, and they'll have different shapes, different configurations. You can see how these binding proteins, how they're going to bind a specific shape is going to fit in there. These are class 1. They're found in all nucleated human cells, which means they're found in everything except red blood cells. Okay. And these are class 1 molecules. Those are the ones that identify self. They don't bind anything. They're not going to bind because if they bound to it, then it cause problems. These are self-recognizing cells. Then the class 2 ones are found in some types of red blood cells. These ones have an antigen that can bind to it. Then it can present it to a T cell to make the proper response. So lymphocyte, which is your white blood cell receptors. So the role of lymphocytes play surveillance and recognition is a function of their receptors. The B cell receptors, okay, they bind the free antigens floating around your system, they bind to it. The T cell receptors, they bind processed antigens together with the HMC molecules on the cell. Okay, they present that are presented to him with the antigen on them. So the antigens are molecules. The chemical structure can vary over a very wide range, billions of different structures and shapes. We recognize them by their shapes. Sources of antigens include microorganisms, bacteria, viruses, and chemical compounds in our environment. Okay. Lymphocytes use 500 genes to produce a tremendous variety of specific receptors. So if all these genes produce these different receptors, could be because of the variety of microbes that are out there. We have the clonal selection theory is undifferentiated lymphocytes or stem cells of the white blood cells in the embryo and the fetus undergo a continuous series of divisions, genetic changes that generate millions of different cell types, each with a very particular unique receptor specificity. So we're binding to certain specificity once they're made, and we can bind to those antigens. So in the bone marrow, lymphocyte stem cells differentiate into either T or B cells. Remember, T or B cells come from the same stem cell. It's just a different pathway it's going to take. Okay? And they both occur in the bone marrow. B cells stay in the bone marrow while T cells migrate to the thymus to become immunocompetent. Both T and B cells migrate to the lymphoid tissues, either the lymph nodes, the MALT tissues, mucus associated lymphoid tissue, or the spleen. Okay. Secondary lymphoid tissues constantly be resupplied with B and T cells. They're always be constantly regenerated and supplied back to these lymphoid tissues. So this is in the bone marrow, and I guess we said it goes one of two ways: B cells or T cells. The same progenitor cell starts it. It just, depending on the stem cell and the conditions, one generates a T cell, one generates a B cell. B cells stay in the bone marrow. Okay, you put these antibodies on the B cells, and then they migrate to the lymph nodes. T cells go to the thymus. From the thymus, they get their T cell receptors on the side of them that are going to look for antigens, and then they go to the lymph nodes. So they're both going to end up in the lymph nodes. The proliferative stage of lymphocyte development, the lymphocytes, okay, specificity is genetically pre-programmed even before an agent has entered your system. In the fetus, in embryo, okay, we start making these B and T cells that are going to run our immune system. They're going to identify these different antigens even before we're exposed to them. So we clone each genetically different type of lymphocyte expresses a single specificity. It clones it, and each one is very specific for what antigen it can bind to. Okay, this is the proliferative stages. Okay, you have the antigen independent period where you're making these without the presence of an antigen. Okay, so we have these lymphocyte stem cells, comes and it starts producing these cells with receptors on it, and the different types of receptors. Okay, and some of these are self. If it's finds self ones, it does not make receptors on that one. Right? But you end up with a bunch of lymphocytes with different, see all the different shapes right here? There's square, that's almost a triangle. That's a little different, that's an arrow. You end up with B and T cells with different specificities, in which and you get all kinds of combinations. And once these are cloned, you have an army of these type or an army of this type, and those circulate through your blood and trigger a response. Then you get antigen that's antigen independent. There's no antigen present at this time when it's developing. Now, antigen dependent, okay, is when an antigen is present. So you have the entry of an antigen, the antigen binds to one of these sites, and then this cell is mass produced. So you get a whole bunch of these cells that have that same specificity for that one antigen being produced. So that's the difference between antigen independent period and antigen dependent period when they're making the T cells that are required for the immune response, T and B cells.
So the dependent one is made when you're exposed to the pathogen.
Correct. When the pathogen is present and it triggers the response. See, in the independent one, you're making a whole bunch of different types. The dependent phase, you're only making one type. That's specific for that antigen. Does that make sense?
Yes, thank you.
Okay. Oh, no problem. Perfect. All right, so the antigen independent period we just talked about. During development of lymphocytes from the stem cells, a given stem cell undergoes rapid cell division to form numerous progeny, whole bunch of different types of antigen receptors. During early cell differentiation, there's random rearrangement of the genes that code for cell surface protein receptors. So you get different variations and different modifications of that receptor shape. This results in a large variety of genetically very distinct cell called clones. Each clone bears a different receptor that would have only a single type of foreign molecule or antigen. No antigen is present, making a whole bunch of different types of B and T cells. Okay. At this time, any lymphocyte clones that carry a specificity for a self molecule and could be harmful or eliminated from the pool of diversity. Okay. So if it identifies it as a self molecule, okay, a receptor that's found on cells in your body, it eliminates that from the pool. It eliminates that receptor. So you don't identify your own cells as for it. So the specificity of a single antigen, okay, is programmed into the lymphocyte and is set for life of a given clone. So that one cell is set for your life. You can have that one antigen with specificity on it. So this results in an enormous pool of mature but naive lymphocytes. They don't know what they're doing yet. They haven't found any antigens, but we have a whole bunch of them. Okay, they can further differentiate under the influence of their home organs and an immune stimulus. Okay, so the first introduction of each type of antigen in the immune system selects a genetically distinct lymphocyte, one of the clones. Okay, one of the clones of those white blood cells that were produced is going to bind to that specific antigen. Okay. This contact causes the clone to expand through mitotic division, okay, into a larger population of lymphocytes that can react to the antigen. So that's a single specificity. Once that antigen is introduced, then you get that one clone responding, just in this case right here. So we get all these different clones, and then the one that binds to an antigen, once it's exposed, then we make more and more and more and more of that one. So we can respond to the attack. Antigen dependent period. We just talked about what happens. Lymphocytes migrate to lymphatic organs, like the lymph nodes. Okay, they enter a specific antigen, enters, and then it binds to one specific clone, one specific type of lymphocyte. It matches the one surface receptor. It's very specific. And this triggers your immune response. Okay, and it's going to vary depending on which type of lymphocyte was stimulated. So all lymphocytes, true or false, all lymphocytes have the same receptor specificity.
False.
False. They all have different, they're all very individually specific. Very good. All right, immunoglobulins. I'm trying to make antibodies now. So immunoglobulins, okay, those are large glycoproteins that serve as specific receptors of B cells and as antibodies. This is where antibodies come in. They're composed of 4 polypeptide chains, and they have a Y shape. There's 2 identical heavy chains, okay, and there's 2 identical light chains. Okay. You have a wide range of antigen binding sites at the end of this fork. They bind right here onto the ends of the Y, okay, due to the variable regions. The constant regions do not vary. In other words, this region right here, these 2 heavy chains, that doesn't vary from antibody to antibody. This part varies between each one degree of specificity, the lighter chains. So this constant part here doesn't really vary from antibody to antibody. This is the end that varies, because this determines the shape or the receptor is going to, what the receptor is going to bind to there on the antigen cell membrane. So only the ends of the light chains vary. Okay, they're held together by disulfide bonds, sulfur-sulfur bonds, just like other ones are proteins, and they form this Y shape. All right. So an undifferent lymphocyte, okay, before it's been exposed, okay, there's about 150 genes that code for the variable regions of light chain, okay? About 250 genes for the variable and diversity regions of the heavy chains. Only a small number of genes for the constant region and the joining regions that links in the final code. So most of your genes are for the variable region, which makes sense. Those will be different. Okay, there's only a small number of genes that code for the constant region because it's constant. You don't need that many genes for it. It's going to stay the same, but your ends are going to be very different to bind to different antigens. So after development, are active in your cell, and all the other variable genes have been deleted. So you get specificity. You only select the one that's going to be used. This maintains lymphocyte specificity. Make sure everything stays specific. It makes sure one thing doesn't bond to multiple antigens. So once it's synthesized, this immunoglobulin or antibody is transported to cell membranes and inserted there to act as a receptor, and it looks for things that are foreign. Okay. So here's your antibody. This is your different variable region right here. This is your constant regions. It's been the same thing on the other side. Right? This is just the DNA that codes for it. Turns it into protein. That's what the immunoglobulins are. Okay, so the heavy chain genes are composed of 4 separate segments: V, D, J, DNA translated into a heavy polypeptide chain, translated by the ribosomes we talked about to make our proteins. The light chain genes are put together like the heavy ones, except the final gene is spliced from 3 gene groups, V, J, and C, making smaller polypeptides. That determines your specificity. So during your final assembly, the heavy and light chains are bound, then the heavy light combinations are connected to form the immunoglobulin molecule. Okay. So it's formed by genetic recombination, just like B cell receptors, with variable and constant regions. There's 2 parallel polypeptide ends. So those are the B cells we talked about. These are the T cells now. So the T cells are very similar, They're going to have a constant region and they're going to have a variable region that identifies the antigens on the T cells. So instead of a Y, these are two. So B cells have the Y antibodies and these have the parallel polypeptide chains. So unlike B cell receptors, T cells are small and they're not secreted. They don't circulate in the bloodstream like antibodies do recreated from the B cells. You stay on the membrane for the T cells and they identify foreign materials there. And you're gonna have two different, you got CD4 and you have CD8. We'll talk about those in a little bit as we go along here. Okay, those are your antigen receptors for T cells. So maturation is directed by the thymus gland, which we talked about, and its hormones. Different classes of T cell receptors, termed CD receptors, or clusters of differentiation. You have CD4. CD4 is one of the receptors found on helper T cells. Those are the ones that bind to the antigen and present it to the B cells that help make the memory. Okay. CD8 is found on cytotoxic T cells. Those are the T cells that actually destroy or engulf the antigen or the attacking microbe. So mature T cells migrate to lymphoid organs. Okay, mature B cells do the same thing. They conglomerate or they accumulate in lymphoid organs. Now the T cells, remember that the receptors stay on those cells, so they're not in the plasma circulating. The B cells make the antibodies that circulate through the bloodstream. Okay, but the cells themselves migrate to the lymphoid organs, both B and T cells. Again, the lymphoid organs are your lymph nodes, your spleen, and your mold, your mucus-associated lymphoid tissues. Okay, so directed by the bone marrow sites that harbor these stromal cells, these stem cells, they nurture lymphocyte stem cells and provide hormonal signals. Millions of distinct B cells develop, and they're called home to specific sites in the lymph nodes, the spleen, and the gall. Okay, they come in contact with antigens throughout your life. They have immunoglobulin as surface receptors for these antigens. So these B cells come in contact with all different antigens as you go through life in the different environments, and they make these different antibodies to the antigens that it's being exposed to. Okay, this is just the difference between T and B cells. Site of maturation or immunocompetence, thymus gland, bone marrow. They both start off in the bone marrow. Markers are T cell receptor CD, okay, MCH1, and then for B cells, it's immunoglobulins, the IgGs, the antibodies. Okay, circulation in the blood for the T cells, high numbers of T cells, low number of B cells circulate in the blood. T cell receptors, TCR, and immunoglobulins are B cells. T cells have receptors that stick on the sides of the membrane. Okay, distribution of lymphatic organ, inferior particles. These are into the middle part of the lymph node. This is on the outer part of the lymph nodes, the cortex. Okay, requires antigens. When presented, yes. B cells, they do not require the antigen because they start the immune response. They're the ones that bind to it and determine if it's going to continue with the response. Okay, you get helper T cells, cytotoxic T cells, and memory T cells. And then for the B cells, you have plasma cells and memory cells that circulate. Okay, so the T cell's basic function is to regulate your immune function, kill foreign infected cells, and synthesize cytokines, which stimulate other reactions for the immune response. For the B cells, they produce antibodies that target, inactivate, and neutralize the different antigens. All right, so an antigen, we talk about that. An antigen is a substance that causes an immune response. Okay? Antigenicity is the property of behavior as an antigen. What makes the human size, its shape, its accessibility, how you can bind to it. All those things determine how strong the antigen is going to get a response from the body. Okay? Antigens have many antigenic determinants, what's called epotypes, small molecular groups recognized by your lymphocytes. Okay, so this is your antigens here. Okay, can circulate in the bloodstream. Okay, so this is your different shapes, sizes, and antigenicity. You can see all the different shapes and sizes here of the different types. Epitopes, okay, are on an antigen, and they're the smaller molecules that are part of an antigen. You can see all the different types of epitopes, which are different type of receptors found on the same invading pathogen. Those are called epitopes. Foreign cells and large molecules, larger molecules, about 10,000 molecular weight, which are pretty large, are mostly antigenic. Complex molecules with several epitopes make good immunogens. They can respond. They have different receptors you can bond to. Poor immunogens, ones that do not initiate a response, okay, are small, simple molecules, okay, like repeating over and over, not attached to a carrier molecule, and simple large molecules like repetitive change of monomers. Monomer over and over is not going to have any kind of different antigens on it to respond to it. If you got a whole bunch of epitopes, you're going to respond real good to it. If it's going to be the same repeating unit over and over and over with not many receptors on it, you're not going to respond to it very well. You're not going to get an immune response as easily. Okay, so here you see that all the different epitopes, and this is your antibody, and it can bind to this one, can bind to that one, can bind to that one. So it can bind to all three of those and bind to that and form an immunologic response. A monosaccharide, a disaccharide, there's nothing to really respond to. They're just repeating units of the same thing, polysaccharides. As long as the monomers are the same, nothing's going to vary on it. And you're not going to respond to it as much as you would a one with epitopes on it and a greater molecular size. Okay. So haptens. Okay. Haptens are small foreign molecules that consist only of a determinant group and too small by themselves to elicit immune response. So it's just a tiny little thing going through the body. You're not going to respond to it because it's too small to bind to anything. Okay. It doesn't fit into anything. Okay. Once it binds to a carrier group, though, carrier groups contribute to the size of the complex. Now, once that antigen or that hapten, which is really small, is an antigen, it's just not like triggers a response. So the hapten binds to a carrier group, now it becomes a larger molecule. Now you can get a response to it. Okay. It enhances how the antigen is shaped, positioned, and you get more immunological response to that antigen. Okay. So these haptens then become epitopes and bind onto this carrier protein. So a hapten is all these small molecules. You inject it, you get no antibody response. Okay. The hapten binds to a carrier protein, and all of a sudden now you got a large area to attack, binds to it, and you get an antibiotic response to it. So the haptens become epitopes, where they're actually attached to the carrier molecule. Okay. We have different categories of antigens. Okay. We have allo antigens. Allo antigens are cell surface markers and molecules that occur in some members of the same species, but not in others. That's why some people respond to bacteria, other people don't. Some people get allergies to different things in the air, other people don't. They're alloantigens. They're different ones that occur within the same species. Determined blood group, major histoincompatibility profile. Okay. AB positive, AB, O negative. They're different blood groups. We can respond differently. Each person, well, not each person, but different people of the same species are going to respond differently because the antigens are different for the different blood groups. Okay, those are alloantigens, responsible for incompatibilities in blood transfusions and organ transfusions, transplantations. Okay. Superantigens. Superantigens are ones that are potent T cell stimulators. They come in, they stimulate T cells, and they activate the whole system. Okay. Now, sometimes those can activate everything, and then the whole system gets activated, and we can't stop it. It's actually like toxic shock. Okay. Massive release of cytokines leading to cell death. And they're healthy cells too. So toxic shock is going to destroy your whole body. Your system is going to shut down. Those are from superantigens. Allergens are antigens that evoke an allergic response. Autoantigens are molecular tolerance is inadequate. In other words, tissues initiate a immunologic response and start destroying our cells, autoimmune diseases. So there are 4 different functional categories of antigens. So which of the following would be least likely to produce a large immune response? A complex antigen, a hapten bound to a carrier molecule, a large molecule with repeated chains of monomers, or whole cells of microbes, whole cells of bacteria.
Is it C?
I would agree with you. It would be C, because I mean, look at your choices. The first one is a multiple epitopes, whole bunch of antigens to bind to. It's complex and you got a whole bunch of things around it. It's going to produce a response. Not likely. A hapten is very small, but it's bound to a carrier molecule. Once it binds to a carrier molecule, then it becomes an epitope and it initiates a response. Whole cells of micro, you got the whole bacteria membrane to produce all these different receptors. Now, this large molecule with monomers, the repeating units over and over and over, don't have many antigens on the surface. It's a single repeating unit over and over and over. Okay, so it would be C. It would be the least likely to produce a large immune response. Could it? Yeah, but it's the least likely to do it of all of those. Okay, so the basis for most immune responses is the encounter between antigens and white blood cells. Those are your lymphocytes. Lymph nodes and the spleen concentrate these antigens and circulate them so they come in contact with the lymphocytes. Okay. So T cell dependent antigens must be processed by phagocytes called antigen presenting cells before their contact with T cells. In other words, you've got to present this antigen to your immune system before the T cells become activated and destroy it. You want to present it to say, look, is this something we should kill or something we should leave alone? Because you don't want to kill your own cells. So you have what's called antigen presenting cells. 3 cell types can serve as these antigen presenting cells. It could be either macrophages, which engulf it. It could be the dendritic cells we talked about that have all these branches coming off of it, off the cell that can trap the antigen, or it can have the B cells. B cells can trap it and present it. So macrophages, dendritic cells, or B cells can serve as antigen presenting cells. Antigen presenting cells modify the antigen. Then the antigen is moved to the surface and bound to what's called an MHC receptor, class 2 receptor. And the class 1 receptors are for self, class 2 receptors are for foreign bodies. Okay. So the antigen presentation involves a direct collaboration among an antigen presenting cell and a helper T cells. That's where your helper T cell comes in. Okay. Now, interleukin 1 or interleukin 2 is secreted. Interleukin 1 is a cytokine secreted by the antigen presenting cell to activate more helper T cells. Interleukin 2 is a cytokine that's produced by the helper T cells to activate B and other T cells, and this triggers your immune response. Okay, so cell activation, you get your foreign microbes, and this is your antigen presenting cell. This is a macrophage, see how it engulfs the cell. So it presents that cell with an MHC2 receptor on the side, and then it's presented to a T cell. There's receptors in there. If it recognizes foreign, interleukin 2 is produced, which activates B and T cell production, and it causes your immune response. And here's what the complex looks like when it's presented. If it's presented and identifies it as foreign, it binds to it, then it recognizes foreign and starts producing T cytotoxic T cells that are triggered to bind to this antigen on the microbe and kill it. Okay. So cell activation, somewhat we just saw in that picture. Here. Antigen presenting cells. On the picture we saw, it was a dendritic cell, all those branches on it, are found in large numbers in lymphatic tissue. Lymphatic tissue is where everything occurs, where immune cells are located, where they frequently encounter complex antigens such as microbes, like bacteria. The antigen-producing cells engulf the microbes, take them into the intercellular vessels, and degrade them into smaller, simpler peptides. The antigen peptide complex with the MHC2 receptors, okay, they're transported to the membrane of the antigen-presenting cell. And from there, location of the antigen readily presented to a T helper cell. Okay? T helper cell is specifically the antigen being presented. Okay? So the APC and the T helper cell cooperate in the formation of a receptor complex that triggers the T cell activation. Okay, so this complex is the HMC2 antigen and the T cell receptor. Okay, so a co-receptor of the T cell after that, the CD4, remember those are just the 2 parallel proteins. It hooks into position on the MHC2 receptor and ensures that the simultaneous recognition of the antigen, it recognizes its non-self, and the HMC receptor as self. So the MHC2 receptor triggers for the non-self reaction. Okay, so the stimuli provide a signal that's related to the T cell genetic material. So it activates the T helper cell. The activated T cells stimulate to release the interleukins to assist other lymphocytes in their function to combat the antigen. Okay, so T cell responses can be cell-mediated immunity. Cell-mediated immunity requires direct involvement of T lymphocytes in the immune response. The T cells secrete cytokines that act directly on other cells. Now, before the T cells get activated, the antigen must be presented and associated with an HMC complex on an antigen presenting cell to ensure recognition of self, to ensure it's the right antigen. We're not destroying our own tissue. Once activated, the T cell transforms in preparation for mitotic division to become a subset of effector and memory cells. Okay, differentiation will be determined by the interleukin that is secreted, whether it's number 1 or number 2. Okay, this is a summary here. It characterizes our subset of T cells. This is a T helper cell 1, T helper cell 2, regulatory cell, and cytotoxic cell. These are the receptors where there's CD4 on the surface, CD4, and the cytotoxic one is CD8. That's the one that causes a destruction of the target cell. This destroys the target foreign cell by lysis. Okay, it's important in destruction of cancer cells, virus-infected cells. Graft rejection requires HMC1 for function, may have some regulatory function. The helper cells activates other CD4, CD8 cells. Okay, interleukin, it secretes interleukin cytokines. Okay, the helper cell 2 drives B cell proliferation, helps in secretions of other cytokines, and the regulatory C cells, development of immune tolerance. Okay, suppression of pathogenic response, inflammation, autoimmunity. It helps turn the immune system off when you need to shut it down. If you turn something on, you're able to turn it back off again once the threat's gone. You can't just keep producing over and over and over again. And you got to be able to turn it back off again. That's what the regulatory C cells do. Okay, so the helper T cells, they express CD4 receptors. Okay, and they're activated by the antigen HMC2 complex. Most of your T cells in the blood and lymphoid organs. They regulate immune reactions to antigens, including other T and B cells. They're involved in macrophages and increasing phagocytosis. Okay. Differentiation depends on what set of cytokines is released from the antigen presenting cells, whether it's going to be helper cell 1 or helper cell 2. Okay. Cytotoxic T cells, these have the CD8 on their cell membrane. Okay. And they also activate the antigen HMC1 complex, and they destroy foreign and normal cells. They secrete perforins and granzymes, which basically dissolve or eat away the offending antigen, offending pathogen. Natural killer cells we talked about before. These guys lack specificity, but they circulate through the spleen, the blood, and the lungs, and they look for things that are foreign, and they destroy them just on their own. No B cells, no T cells, they just destroy. Okay, and they can destroy cancer cells and tumors that we have no idea how they're identifying it. It's kind of a surveillance system in the body. So we talk about the T cell activation. Okay, here's an antigen presenting complex on this dendrite. Okay, you get cytokines released that causes T cell to differentiate. And it can go to memory cells. It can go to the produce a macrophage. Okay, and it goes into the B cells and activate the B cells to produce memory antibodies, increases your inflammatory response. Okay, and then the regulatory T cells decrease it once the threat's gone. They decrease the immune reaction. Okay. Same thing on the other one. It differentiates the T cells, but this activates to the cell recognizes the infected self of it. Instead of going through all the different processes, it activates it directly. Then you get granulocytes produced, perforins produced, and these secretions from the cell act to destroy the cell. So the T cell itself doesn't destroy it in this response. It causes the cell to produce perforins and granulocytes that will destroy the infected cell. This is a scan electron microscope. You can see the cytotoxic T cell. And this is a cancer cell. This is the dead cancer cell. And this is a toxic T cell. Kind of cool pictures. Natural killer cells. It finds a foreign cell. It releases perforins and granulozymes. These perforins and granulines go in and they actually punch holes in this, and you get lysis of the cell, and it kills the cell. So NK cells, natural killer cells, release perforins, which polymerize and form a hole in the foreign membrane, and granzymes, okay, enter the perform hole and degrade foreign cell proteins inside. Once the hole is made, these perforins enter it, okay, and destroy the cell from the inside. So the foreign cell dies by apoptosis, by destruction of the internal organs, internal parts, and the macrophage engulfs and digests the dying cell. And this is a picture of a natural killer cell, an NK cell. Okay, so the reaction has drastic consequences. T cells and T cells and superantigens. Okay, so superantigens are found primarily in bacteria and viruses, and are a form of a virulence factor when they're very strong. Enterotoxin from pathogenic staphylococci, certain toxins of group A streptococci, and proteins of the Epstein-Barr virus. They provoke an overwhelming immune response by a large number of T cells, regardless of specificity. So these superantigens stimulate a tremendous immune response. We get release of massive amounts of cytokines. We get blood vessel damage from that release. We get toxic shock where organs start to fail, and then multi-organ failure. Because the immune system gets basically turned on and we can't shut it down. It just gets turned on maximum amount and it just starts destroying everything. Okay. So the lymphocyte responsible for recognizing and destroying cancer cells is which one? The antigen presenting cell, the B cell, the cytotoxic T cell, or the T helper cell?
Is it the cytotoxic cell?
The antigen for recognizing and destroying. Yeah, because I recognize it. It says destroying. The only one that destroys it would be the cytotoxic T cell. Correct. That'd be perfect. Yep. Events in B cell responses. So B cell activation and antibody production. Remember, our B cells are responsible for producing the antibodies, which serve as our memory. So once B cells process the antigen, they interact with the helper T cells and are stimulated by growth and differentiating factors. They enter the cell cycle in preparation for mitosis and clonal expansion. They produce more of that one type of B cell that's going to identify that one specific antigen. Divisions give rise to plasma cells that secrete antibodies and memory cells that can react to the same antigen later on, so we can recognize it again. So the main events involved in B cell responses are 6 steps. Clonal selection, it binds to the antigen and makes that one specific antigen. Induction by chemical mediators, the cytokines are released and they determine what type is going to be produced. B cell T helper cell cooperation by recognition. B cell becomes activated, clonal expansion, that one type of B cell is produced now over and over. And then antibody production. And once antibodies are produced, they are secreted into the lymph, into the bloodstream and circulate. Okay. This shows our B cell activation. Okay, we'll go through this step by step. On the next slide here, you get clonal selection and antigen bonding. You get antigen processing and presentation. You get the B cell complex with the T helper T cells and recognizing it as foreign. The B cell becomes activated. Then you get clonal expansion for that memory cell. These memory cells expand and you produce more of these. And then you get plasma cells, which produce your antibody in response to that one antigen. So this breaks it all down. Clonal selection and antigen binding. B cells can independently recognize microbes, okay, like a virus in this case here. Okay. And they're foreign antigens and combine them with the immunoglobulin receptors. Immunoglobulin receptors are what determine your antibodies. This is how the initial selection of antigen-specific B cell clone occurs. So it's triggered by that one specific antigen. Antigen processing and presentation. Once the microbe is attached, The B cell endocytoses it, brings it inside, processes it into smaller protein units, and displays these units on the outside of the membrane. Displays them onto the MHC2 complex. Okay, this then readies the antigen for presentation to a specific helper T cell. Then you make a helper T cell B cell complex to recognize it as foreign. Okay, for most B cells to become functional, they must interact with a T helper cell that bears receptors for the antigen for that same micro that's being presented to the outside on its membrane. Okay, this T cell may become activated by the antigen presenting cell. The two cells engage in linked recognition, which the MHC2 receptor bearing antigen on the B cell binds to both the T cell antigen receptor and the CD4 molecule on the T cell. Remember, the CD4 molecule is used for recognition. The CD8 is used for destruction of the cytotoxic T cell. Then you get B cell activation. So the T cell gives off additional signals in the form of those interleukins and the B cell growth factors. The linked receptors and chemical stimuli serve to activate the B cells. Okay? Such activation signals increase in cell metabolism, leading to cell enlargement, proliferation, and differentiation. So differentiating that one specific receptor for that antigen. Then you get clonal expansion, memory cells. Goes to one path goes to the memory cells of the B cells. The other goes to more expansion to produce the antibody that's going to bind to that foreign material. So the activated B cell undergoes numerous mitotic divisions, which expand the clones of cell bearing the specificity and produce memory cells and plasma cells. So it produces memory cells, going to remember it. So it's going to have, that was good, this whole process again. And has plasma cells going to produce the antibodies. Memory cells are persistent long-term cells that can react with the same antigen with future exposures. That's how vaccines work. Okay. So plasma cell antibody synthesis. Plasma cells are short-lived. So the memory cells are the long one, they'll produce the antibodies, but those are long-lived. The plasma cells are short-lived. Okay. They actively secretory cells that synthesize and release these antibodies into your bloodstream. These antibodies, these are IgM ones here, there's 5 different types we go over, have the same specificity as the immunoglobulin receptor and circulate in the fluid compartments of the body where they react with the same antigens and microbes that were originally presented to the cell. So now we got antibodies. Remember, antibodies were that Y-shaped. Well, one of the IgG was a Y-shaped antibody, where you have your binding site and your constant sites. So immunoglobulins, okay, with 4 polypeptide chains, the 2 heavy and the 2 light. Okay, and they're connected by disulfide bonds, sulfur-sulfur bonds. All antibodies have 2 functionally distinct segments. The antigen binding fragments called Fabs, which are in the arms. They're amino terminal ends, variable regions of the heavy and light. Those are the antigen binding sites, these Fabs. Crystallized fragments are the FC. These are the constant sites. They bind to various cells and molecules of the immune system. Okay. This is your constant site. This is your FC. This is your Fabs up here. This is where the antigens bind. And this is your Y-shaped antigen. Okay. You can see the antigen binding sites. Okay, this is the Fab portions of it, and then these are the heavy chains that connect, the 3-dimensional images of it. Okay. So the Fab antigen binding site is composed of hypervariable regions, because we have extreme variable, because this is the region, this is the area where it's going to attack to the different shapes of the antigen. So it has to be very variable. It has to be extremely variable, hypervariable. Okay, extremely variable amino acid content because it's going to be different coding from the gene. This groove of the antigen binding site has specific 3-dimensional fit for the antigen. The specificity of the 2 Fab sites is identical for each antigen. Okay, so when they come together to make the 2 sites, they're going to match that exactly. In other words, you look at this here, these are going to match perfectly for binding. One's not going to be square, one's not going to be round. Those have the exact same shape for binding. So an Ig immunoglobulin molecule can bind antigenic determinants on the same cell or 2 separate cells, and then link those 2 together for identification of the antigen. Okay. And again, you can see how it binds to both parts here. And this is your hypervariable region of the Fab. And that's a good fit. You see how it fits right into it. Okay. This is a poor fit, or actually no fit at all. This is a poor fit. Parts of it bind, but most of it doesn't for the antigen. Okay, so this will give you a good immune response. This will give you no immune response. This will give you a partial immune response. Okay? So what do antibodies do? Antibodies are antigen. They unite with, they immobilize, they call attention to, or they neutralize the antigen from which the antibody was made from. Opsonization is a coating of the microorganism or other particles with specific antibodies so they're more easily recognized by the phagocytes. Okay. So opsonization is when the antibodies circle the blood, see the antigen that they've been trained to identify, bind to it, and then they coat this antigen with all these antibodies. The macrophages see all these antibodies, go, we need to destroy the cells being found as foreign, and they dissolve it. Neutralization, antibodies fill the search receptors on a virus or active site of the microbe enzyme to prevent it from attaching to a healthy cell. They just don't allow, they don't necessarily kill it, but they neutralize it. They don't allow it to connect to a cell so it can't propagate itself. So opsonization, they mark it and it gets destroyed. Neutralization is they bind to it and they prevent it from attaching to other cells. Okay. Agglutination. Agglutination was the antibodies aggregate on the cell. They come together and they form into large clumps. Okay, so these antibodies from cell to cell to cell, and they stick to each other, and they form an agglutination, or a big clump. Complement fixation is activate of the classical complement pathway, results in specific rupturing of cells and some viruses. Okay, it binds to it, causes a hole in the membrane, causes leaking of the cell. Precipitation is an aggregation of particulate antigen. In other words, it takes the antigen and it forms a mass of these sticks them together. It's such a large mass, it forms what's called a precipitate, which is a solid. So this is complement fixation. It causes lysing of the cell. Okay, antibodies bind to the surface, activate the complement system, lead to lysis of the cell and some viruses. It kills the cell. Opsonization. Opsonization, all these antibodies bind to the cell and they mark it. See all those little markers on the cell? Now the macrophages recognize those markers and they say, oh, this is a foreign cell because it says so, because all these antibodies are on it, and it dissolves it. Neutralization is the antibodies bind to it and the cell cannot enter, or the virus in this case, cannot enter another cell, so it neutralizes it. Okay, agglutination is the antibodies link with one another and form this big, like a mass here, and prevent it from going anywhere. Okay. Precipitation is the solids actually formed, but all these antibodies bind together. They form precipitate. And then antitoxin is that toxins are released. Okay. Toxins bind to it and destroy the antigen. Okay. Antibodies can be used to bind to bacterial endotoxins, but bacteria can release endotoxins also. So these antibodies can bind to that, okay, intercepting them before they have a chance to cause cell damage. Okay. So remember, FC are the constant fragments. They bind to cell membrane. These are your macrophages, neutrophils, eosinophils, mast cells, basophils, lymphocytes. Those are all your different lymphocytes. Regions on the FC portion, the constant portion of certain antibodies, fixed complements. Okay, the binding of these consistent sections cause release of cytokines. Okay. So this is your complementary sections right here. Again, this is the same picture with the three-dimensional representation of them. We have 5 functional classes of Ig, immunoglobulins. We have IgG, IgA, IgM, IgD, and IgE. IgG is a monomer. So it's only one Y-shaped antibody produced by plasma cells. It's your primary response, and memory response is secondary to it. And it's the most prevalent antibody we have in the body. IgA is another monomer. It circulates in the blood, and it's a dimer in mucus and serous secretions. It becomes 2 instead of the 1. IgM is 5 monomers that connect together, synthesized by following the first antigen encounter. IgD is the monomer, serves as a receptor for antigens on the B cells. And IgE is involved in allergic response and in parasitic worms and infections in the body. Now, these variations are due to the constant portions of the immunoglobulin. So this is IgG. You can see it's a monomer. This is IgA, which is a monomer, and when it's secreted in the secretory tissues, it becomes a dimer. with two of them together. This is IgM. IgM is 5 antibodies connected together. IgD is another monomer. And IgE is another monomer. You see, they all have different functions because the different amino acids arranged on their constant parts on the FC portion. All right, so different characteristics of a monomer, a dimer, a pentamer, and then the monomer, IgA, the different Ig classes. When it's only a monomer, you only have 2 binding sites. When it's a pentamer, you have 5 of them. There's 2 on each one, you have 10. Okay, molecular weights in a monomer is like 150,000, and it can get to 385,000 for the dimers, which is doublet, or it can go to 900,000. Okay, for the pentamer, which is 5 of them. Percentage of total antibodies here, 80%. So 80% of your antibodies are IgG antibodies. These are the monomers. Okay. Average life in the serum is like 23 days for the IgGs going. The other ones are much shorter lived. Okay. Do they cross the placenta? The IgG does and imparts immunity to the fetus. The other ones do not. Fixes a complement. Okay, binds 2 different phagocytes, epithelial cells, and the biological function. These are your long-term immunity. This is your memory antibodies. They neutralize toxins and viruses. Okay, the dimer or the monomer secretes antibodies on the mucous membrane, and the pentamer, the IgM, okay, produces the first response to antigens, can serve as B cell receptors on it, and then the monomer and dimers, these are in very small amounts. Receptors on B cells and antigen recognition, antibodies of allergy. These are worm infections, parasites. Okay, so antigen binding sites of an antibody are located in the light chains alone, heavy chains alone, FC region of the antibody, or the Fab region of the antibody. Where is the antibody antigen binding site?
Is it C?
The FC is the constant region. Those are the body. This is your FC parts right here. So I'm drawing that backwards.
Yeah, I was gonna say C as well, but I don't know.
It would be in the Fab region. This is your Fab region where you're binding on the outside. Remember, your antibody comes in here. Wait, it's a hard time drawing this. This is your Y shape. Okay, and these determine your antigen binding sites. Okay, this is your FC region. This is a constant region. Remember, C is for constant. Okay, this is a variable region. This is where the different antigens bind to it. So it binds in. This is your Fab region. This is your FC region. So the Fab region is where the antibody is actually going to bind to it. The antigens are going to bind to the antibody there. Those are your binding sites. So it would be the Fab region of the antibody. Antibodies in the serum, circulating in your bloodstream. So regardless of the site where antibodies are first secreted, okay, a large quantity eventually end up in the blood. So an anti-serum is a serum that contains specific antibodies. You can separate the antibodies by electrophoresis, like you do with DNA, by different globulins in the anti-serum, separate into 4 bands using electrophoresis. Electrophoresis is where you apply a charge to a sample, and the larger proteins move slower. The smaller ones move quicker. They're pulled faster. That's where you separate DNA into also. We get alpha 1, alpha 2, beta, and gamma. We get those 4 different globulins, immunoglobulins in the serum. Most of these are antibodies. Gamma globulin, which is our Y shape, is most common. That's the IgG. That's 80% of all our antibodies. Okay, beta and alpha gamma globulins are a mixture of IgG, IgA, and IgM. This is your primary response. This is 10 to 12 days. You get your IgM response and your IgG response. It takes about 20 days, so roughly a week and a half to 2 weeks to build a response, and you start to feel better. Now, second exposure, look at the difference. About 3 days, you start building a response. You get an IgM, you get IgG, tremendous response here. This is a result of vaccination or exposure to the antigen previously. Okay, so in 5 days, you basically got to get high levels. Look at the difference in the level. The levels here and the levels here, and only like 5 days. So you kick it out before it has a chance to respond. You kill it quickly. That's the whole reasoning behind vaccines and our whole memory system for immune system. We can respond quicker to the second exposure. much quicker and much greater response. Everything's already made. So all we gotta do is just dump it in there and produce those Ig ones from the memory cells that are already there. So they find it, recognize it, and just dump our system with it. That we would kill it off really quick. And this other one, you don't get that big response, and still, 25 days later, you get your maximum response. Here we get our maximum response in less than 10 days. So the primary response, okay, this is a latent period with no measurable antibody occurs early on because you have to form it. We have to get the recognition. We have to find the recognition. We have to produce the memory cells. We have to figure out the T cells. We have to get all those set for that particular antigen on our first exposure to it. Okay, so we have this latent period, just our body's working to try to fight it. So the first antibody to appear is IgM, followed by IgG, which arrives from activation of the first memory cells. Within weeks, it tapers off to low levels. Second response. Now, this is we've already been responded to, or we've been exposed to it, whether intentionally or non-intentionally. So a latent period is lacking because other memory lymphocytes from early responses are immediately ready to react. You don't have to make all those this time. We're already there. All you do is take those memory cells and dump our body with these antibodies from it. So a rapid response and an antibody titer, mainly IgG, that's our big one. It's the same for several weeks. Small amount of IgM is also produced by the naive B cells. So we monoclonal antibodies, the ones that are cloned from one particular antibody, they originate from a single clone, like we talked about, and have single specificity for an antigen. It's a pure preparation of antibodies. There's a single specificity antibody formed by fusing a mouse B cell with a cancer cell, okay? Used in diagnosis of disease, identification of microbes, and therapy. Okay, we'll show about these monoclonal antibodies. This technique is producing monoclonal antibodies by hybridizing melanoma tumor cells with normal plasma cells. So newer MBAs take advantage of mice that have been genetically engineered to develop a human immune system. So we take mice that have, and we can alter their immune system. So these are antibodies we can use for treatment. We can make these antibodies, okay, in the lab so we can help with Herceptin, like with breast cancer. We can make specific antibodies for it, which we have done now, Herceptin. Okay, leukemia, lymphoma, CAMPATH, metastatic melanomas, lung cancer, okay, asthma, ulcerative colitis, Crohn's disease, respiratory syncytial virus, rheumatoid arthritis. We can make these in the lab using this technique of monoclonal antibodies to help fight the infection if your body's not fighting the infection on its own. Okay. So these categories we can create. We have active immunity, we have passive immunity, we have natural immunity, and we have artificial immunity. These are different ways to become immune and use these antibodies I produce in our system. So active immunity. So when a person is challenged with an antigen, stimulates the production of antibodies. I think I got a chat message here.
I don't know if you guys can hear me, but
I can now, yeah.
it keeps logging off for some reason. So I just wanted to let you know, or I didn't know if you could hear me or not, but it keeps kicking me off. So I'm not doing that on purpose. So
no problem.
Making a noise or whatever, that's not me.
No problem, Marissa. Megan had that same problem last week. Are you having the same problem this time, Megan?
No, but that did happen to me last week too.
Right.
Yeah.
I think it's when there's storms out in the desert area, it really affects the internet down there. I think that's what it does in different parts. So it's not a problem at all.
Yeah, it's just being really.
Yeah, I'm used to it.
All right. I just
So
wanted to let you know.
Not a problem. I appreciate it. Not a problem. So active immunity. That's where a person is challenged with an antigen, stimulates production of your antibodies, creates memory, takes time, but it's long lasting. It forms a memory. That's your active immunity. You get an antigen, your body builds up a response to it, takes time to develop, okay? And then it creates memory. Passive immunity. Preformed antibodies are donated to an individual. Does not create memory, but acts immediately and is short term. In other words, you take a patient that is not responding, that gets sick. So then you have another patient that has the antibodies to it. You inject the antibodies from that patient into that patient. Those antibodies kill off the disease, but that doesn't create memory because you're not creating memory B cells, you're just sticking in antibodies. But it does help get that one infection under control. Natural immunity, acquired part of normal life. You're exposed to all these things as a child, and you build up these natural immunities, these natural B cell activations from the antigens in your environment. Artificial immunity is acquired through a medical procedure like a vaccine. Vaccines are injected. What we do with there's certain types of vaccines, we'll talk about this in a little bit here too. Some that are attenuated. The flu vaccine is an attenuated vaccine. It's a dead virus, it's killed. But the outer protein shell is intact. So when you inject someone with that, you don't have the virus, but you have the antigen, which is identified as the coating of the membrane. So you find the antigen, the body responds to it. So next time you're exposed to this virus with a live one out in the environment, the body says, hey, we know this is foreign, and it attacks it so much. Just like those graphs we saw, attacks it so much sooner and quicker. So natural active immunity. Okay, so getting an infection. After recovering from the infectious disease, a person may be actively resistant to reinfection. And a period varies depending on the type of disease that you got exposed to. Because once you got infected, if you survive, okay, you made antibodies to it. Now you got the immunity in your body. So the next time you're exposed to it, you're not going to get it again, by theory. Okay. Natural passive immunity is like mother to child. IgG antibodies from the maternal bloodstream cross the placental barrier into the fetus. IgA antibodies cross are in the mother's milk. Okay. They prevent microbes from entering in the intestine. Artificial active immunization, vaccines, microbial antigenic stimulus, okay, triggers the immune system to produce antibodies and memory cells. Artificial passive immunity is immunotherapy. Patients at risk for acquiring particular infection is an administered preparation that contains specific antibodies that act against an infectious agent. This does not create memory because you've not triggered any of the B cells. This is strictly putting antibodies into your system. The antibodies are already there, and they mark it as bad or non-self, and your body destroys it. But you don't have memory of that when you do the artificial passive immunization. Okay, so acquired immunity is like natural immunity. It's acquired through normal life, experiences not induced through medical means. So it's not a vaccine, it's just when you go out and play as a kid, you get exposed to other kids, you pick up the viruses from them, pick up viruses out in the environment, and you get immune to it. Your body responds to it, and you build up antibodies to that. Active immunity results when a person develops his own immune response to a microbe through an infection. Okay? So in other words, the difference between here and natural immunity, you did not get the infection. You picked up the disease, your body fought it, you didn't even know about it, and it built up antibodies, and you did this. Now, sometimes they'll give you the infection, and then your body fights it. That's an active immunity. It results when a person develops his own immune response to a microbe through an infection. Passive immunity, passed through the breast milk. Okay, passive across the placental barrier. Remember, IgA crosses through the breast milk into the infant. IgG antibodies pass through the placenta. That's called passive immunity. Okay, so that's immunity by another individual. Artificial immunity is produced specifically through medical procedures, immunizations, vaccinations. Active immunity is a vaccination. It results when a person has his own immune response to a prepared microbial antigen. Passive immunity is when occurs given selected immune substance by another individual. You can feed it to them IV. Okay. So artificial passive immunization is useful for immunocompromised patients who cannot be vaccinated. There are certain people that cannot take vaccines. People that are allergic to eggs cannot take the vaccine, the flu vaccine, because it's incubated in eggs. So for these people, you can use artificial passive immunization. Okay, this lasts about 2 to 3 months, the injection of the person of antibodies around the person. You can do human Ansera is the immune serum globulin, ISG. This is gamma globulin, the Ig antibodies. Blood of about 1,000 donors, treatment of choice for preventing measles, hepatitis A, replacing antibodies in immunodeficient patients. So you replace their antibodies. Specific immunoglobulin, okay? These are from donors in a hyper immune state after being vaccinated or infected by like pertussis, tetanus, chickenpox, hepatitis B, and then you can inject that into a patient. Okay. So the anti-sera, antitoxins of animal origins, the sera produced in horses are available for diphtheria, botulism, spider and snake bites. We can produce that serum, okay, those antibody or antitoxins in the horses, and we can inject them into people that have diphtheria or like snake bites. Artificial active immunity can be conferred artificially by vaccination. Deliberate exposure to ensure that it's antigenic but not pathogenic. Okay, that's the key. So vaccination, the material is antigenic, but not pathogenic. Does not cause a disease, but it causes the antigen response. It causes us to make antibodies. The principle is to stimulate primary and secondary anamnestic response to prepare the immune system for full exposure to a viral pathogen. So we get ready for when we expose to the real pathogen, we can fight it. So the response to a future exposure is going to be immediate, powerful, and sustained. We're not going to have to wait on it. So most vaccines, okay, are prepared from killed whole cells or inactivated viruses. Live attenuated cells of viruses. Attenuated means that they cannot reproduce, they cannot function. Antigenic molecules derived from bacterial cells or viruses, genetically engineered microbes or microbial agents. Those are the sources of vaccines. 4 different ways we could produce vaccines. Now, you want the vaccine to be effective. For a vaccine to be effective, we need certain requirements. You can't just inject things into people. It should have a low level of adverse side effects or toxicity and cause no serious harm. It should protect against exposure to natural wild forms of the pathogen out in nature. It should stimulate both B cells and T cells. B cells stimulate for our memory, T cells our response to it. It should have long-term, long-lasting effects. It needs to produce memory cells. It should work with minimum dose or boosters. And it should be inexpensive. We should have it available to everybody. Relatively long shelf life and be easy to administer. Those are requirements for an effective vaccine. Okay. Advantages of live preparations are organisms can multiply and produce infection, but not the disease, like the natural organism. Okay, so the body can respond to it, doesn't get the disease. They confer long-lasting protection because it stimulates B and T cells, and they usually require fewer doses and boosters. Disadvantages require special storage, can be transmitted to other people, can conceivably mutate back to the virulent strain. So in vaccine design, these are whole cell vaccines. We have the kill cell or inactivated virus, okay, heat or chemicals. Okay, this is a dead virus, so the virus is killed inside of here, but you still have these antigens on the surface. Antibodies respond to it, and we get our memory cells, and we get our immune response. A cell, B cells, T cells respond, and it forms memory. Same effect as with the live or attenuated cells or viruses. We still get that same antibody antigen response to produce our B cells and T cells. Vaccines from microbial parts, we take the part of this antigenic surface and break it apart, and these antigens now are free floating. They get incorporated into the vaccine bacterium, and bacterium mass produces these portions of the antigens. These antigens can be injected in. The antigens are not infectious, but they're going to produce an immune response because the antigens that we're going to respond to. Okay, so in vaccine designs, you want exact antigen determinants can be used when it's known, like capsules. We use capsules for pneumococcus and meningococcus. Surface proteins for anthrax, hepatitis B, exotoxins for diphtheria and tetanus. So the antigen can be taken from cultures produced by genetic engineering, or it can be synthesized. We know what specifically what we're dealing with. Recombinant vaccine. We can take our plasmid, put in a DNA we want to produce a certain gene, produce that certain gene is going to produce that antigen, take those antigens, inject it in, and we have a vaccine. Okay, without the presence of the bacteria or the virus. We just have the antigen that's going to stimulate the immunological response. Okay. So I'm going to insert genes for the pathogen into a plasmid vector. Those are the round DNA structures in bacteria, and clone them in an appropriate host. Stimulated cloned host is synthesized to create a protein, which is your antigen. We're going to harvest it and purify this antigen. Okay, it's like the Trojan horse vaccine. You have genetic material from a pathogen, is inserted into a live carrier, which is a non-pathogen, and then it expresses the foreign genes. We try this with vaccines with AIDS, herpes simplex, leprosy, tuberculosis. Okay? We can use these carriers to produce the antigen that we want without causing the disease, without having a viable vaccine virus. Okay, genetically engineered vaccines, you do DNA vaccines, create recombinant by inserting microbial DNA into a plasma vector. Human cells pick up the plasmid and express the microbial DNA as proteins, causing these B and T cells to respond, be sensitized, and then we form memory cells from it. Okay, so we take this plasmid from the bacteria, introduce it into human cells, and we can produce the antigens that we need so the body can respond to it. We have experimental vaccines for Lyme disease, hepatitis C, herpes simplex, influenza, tuberculosis, and malaria so far in doing this. So this is how we do it. We take the DNA of the pathogen, we inject it into the plasmid of the bacteria, take that, put it into the person. This plasma gets incorporated into the DNA of our cells. And then once it's in our DNA, it can express the genes that we injected into it and produce these antigens. These antigens can then trigger an immune response. So when the live virus comes in with these antigens and attacks into the body, it has an immune, we already have memory to it. We can get there and attack it and kill it really quickly. So DNA that codes for a protein antigen is extracted from the pathogen's genome. Genomic DNA is inserted in the plasmid vector. Plasmid is then amplified as we prepare the vaccine. DNA vaccine is injected in the subject. Cells accept the plasmid. DNA is incorporated, transcribed it, into the cytoplasm, translated by the ribosomes into the various proteins. The formed proteins is inserted into the cell membrane, where it's going to stimulate a cell response without having the effects of getting sick of the infection. We're just creating the antigen on the surface of our cells so we can respond to it. And then we can get our second response. We get exposed to it in nature and it can respond quickly. Okay, these are some of our vaccines currently licensed in the United States. Tuberculosis, typhoid, anthrax, meningitis. Okay. Pneumococcal pneumonia, diphtheria, tetanus, hepatitis A, influenza. This is a common cold, flu vaccine, encephalitis, polio, rabies, adenoviruses, chickenpox, influenza again, measles, mumps, other rotavirus, rubella, smallpox, and yellow fever. Okay. We also have hepatitis B and human papillomavirus. And we got diphtheria, hepatitis B and A, measles, mumps, rubella, and chickenpox also in that. All right, so those are the vaccines that we have. So only a portion of a pathogen is being used as the basis for a vaccine. It'd be called what? Attenuated, acellular, killed, or recombinant. If we take a portion of the pathogen, a small part of it, and use that for the base vaccine, what do we call that?
Isn't that D, recombinant?
And the recombinant when we put in,
Or
like,
A. Is it A?
attenuate when we kill off the DNA so it can't hurt. It'd be an acellular vaccine, but with no cell with it. We're taking a small portion of it. Recombinant vaccines, we put it in the plasmid ring. The kill vaccine, we destroy the inside. Attenuated is we isolate just the active portion of it. But an acellular vaccine, we take a small portion of it and bring it in. So it'd be the acellular without, in other words, no cell vaccine. Does that make sense?
Yes.
Okay, so that'd be the acellular vaccine. Okay, routes of administration or side effects of vaccines. Okay, most administered by injection. A few can be oral or nasal. Some vaccines require an adjuvant. Okay, it's a component that enhance the immunogenicity and prolong retention of the antigen. Stringent requirement for development of vaccines. There has to be more benefit than risk. That's the big thing with the COVID vaccine right now. Everybody's trying to show, but if you listen to it, you have to prove more benefit than risk of the vaccine itself. Possible side effects include local reaction to the injection site, fever, allergies, rarely back mutation to the virulent strain, neurological effects, Guillain-Barré syndrome, It's a big problem with the COVID vaccine and other vaccines. The bird flu vaccine was one with the Guillain-Barré syndrome, a big side effect neurologically. When known as effective adverse effects, oh, did you say something?
Oh, did you say the gray? Isn't that mad cow's disease?
No, Guillain-Barré syndrome is a neurological symptom from vaccinations. The bird flu vaccine caused a high percentage of it. The COVID vaccine caused a small percentage of it. It's when your neurological system gets destroyed, you have no control of your muscles anymore. You can't walk, you can't function. It's called Guillain-Barré syndrome. I think you're thinking of Jacob Kleinfeld syndrome. That's like mad cow syndrome.
Oh, okay. I think I was confusing those two.
That's right. There's a lot of things out there to get confused with. Okay. Side effects include a reaction we talked about, neurological effects. When known or suspected adverse effects have been detected, vaccines are then altered or withdrawn from the market. Okay. Herd immunity. Immune individuals will not harbor the disease. Reduces the occurrence of pathogens. It's called herd immunity. So people get it, they get over it, they get the antibodies, they don't pass on to anybody else because they removed it from the population. That's how herd immunity works. That's how most of the things we do to fight viruses work. We work by herd immunity. It's less likely that a non-immunized person will encounter the pathogen, because it's been removed from the population, because once you fight it and get rid of it, you don't harbor it anymore. So you don't pass it on.